Hi, everybody. Welcome to the digestive system. This is going to cover the way that we can break down food into nutrients. So components of the digestive system begin at the mouth, go down the pharynx, the esophagus, the stomach, small intestine, large intestine, and then waste would be moved out. Other components would be like the salivary glands, the liver, the gallbladder, and the pancreas, which all create things that get added to the digestive tract to help the digestion process. The function overall is to process and digest food while also eliminating waste from your body. The idea in consuming food is to absorb nutrients so your body can create energy. And then also the digestive system is going to help regulate the nutrient content in the blood through secretions of the liver and the pancreas. Okay, so the digestive system is called the alimentary canal or the GI tract, which basically just tells you that there is a tube that's going to be carrying all this material throughout the body. For homeostatic functions, it's going to be to acquire materials in order to create energy. The organs here of the digestive system, so mouth, pharynx, esophagus, stomach, small and large intestine, and anus. And then the accessory ones, again, were the glands, the salivary glands, liver, gallbladder, pancreas, but also the teeth and the tongue because they help begin the process of breakdown mechanically, which just means physically. So the process of digestion, you have ingestion, which means the intake of food. Digestion is actually broken down into two different components, right? The first one is going to be mechanical breakdown of food. That's going to be physically breaking it down. And then the chemical breakdown of food is going to be absorbing it in from the opening in the digestive tract, which means the lumen, into the body, which means the bloodstream. The mechanical process, again, is going to include mastication, which is chewing of food, and then motility, which just means the movement of it. And there's different types of movement depending upon where you're wanting to move it from and to. And one of them is called peristalsis, and the other one is called segmentation. And then next is secretion. So secretion means fluid and digestive enzymes, acid, alkali, bile, mucus, and hormones are all going to be put into the digestive tract to help the process of breakdown. Absorption again means moving the nutrients from the GI tract into the blood or lymph, just depending on size. And then elimination is undigested or unabsorbed waste that's going to be eliminated out of the body. Okay, so if we start with movement, right, motility, there's two different types. There's peristalsis and segmentation. Peristalsis is going to be the essentially linear movement. So basically moving it in one direction, like forward or down. And in this case, it happens mainly in your esophagus, right? The esophagus is not for absorption. It's just a passageway. So it's going to help move food from the mouth into the stomach. When you put table food into your mouth, you are going to add in saliva. And saliva has a couple different components like water and mucus and enzymes and antibodies and ions. And because you're adding all of that to it, when you put table food into your mouth, you don't actually call it food anymore. You're going to call it a bolus. So peristalsis is going to help move the bolus from the mouth down the esophagus into the stomach. And it's going to be lined with smooth muscle to help the bolus move linearly, or in this case, longitudinally from up to down the tube. The second type of movement is called segmentation. This is going to be the breaking up and mixing of the bolus. This is essentially like a churning, right? You're turning over the food. And these are going to be random contractions, primarily occurring in the small intestine where you have lots of circular muscle. So again, this is longitudinal movement, right, from the mouth linearly. In this case, what happens with peristalsis is the bolus will move and then you'll have a contraction occur behind it and propelling it forward. Segmentation is churning. Again, you're going to kind of turn the food over, mix it up, and break it up like this. Okay, so when we talk about layers of the GI tract, there's 4 possible layers. Mucosal layer, submucosal, muscularis, and serosa, or also it's called adventitia. The mucosal layer is basically made up of mucus, and it's an added layer of literally mucus, and it's going to like line the stomach to prevent acid from burning holes in your stomach, right? That'd be like a stomach ulcer. The next layer is going to be submucosal. This is dense, irregular connective tissue. Here you have nutrients that are going to be absorbed into the lymph or the blood, and you also have nervous innervation here. Next is going to be the muscularis layer, which is this layer over here. You have smooth muscle and potentially 3 different types, circular, longitudinal, or even oblique muscle that are going to help contractions occur in different ways. And then the outside layer right there is the serosa, that's connective tissue that's going to be connected to the visceral peritoneum. Okay, so your mouth is going to be the oral cavity. This is the beginning of the mechanical breakdown of food, where basically you use the teeth to chew and the tongue to move food. Then you have the chemical breakdown of food with salivary amylase. Here you're going to have an enzyme which is going to help digest carbohydrates. So when you talk about the macromolecules that we can utilize for the production of energy, right, you have carbs, fats, and proteins. The only one of those that begins breakdown in the mouth is carbohydrates. Then you can also moisten the food with secretions from the parotid, sublingual, and submandibular glands, and that's just like the saliva. And then the next part says mastication, that's chewing and mixing the food with water and enzymes. And then ingestion, meaning you're going to start to move the bolus from the mouth down the esophagus. And then the last part is gustation. This is where you get taste, right, or a special sense of taste. So the mouth, right, is going to be made up of the labia and the vestibule. Then you have the anterior border, which is going to be the lips and the teeth, the posterior border, which are the fauces and the oropharynx, which just means the back of the mouth, and then the fauces is the opening into it. On the superior side, you have the hard and soft palate. And then on the inferior side, you have the mylohyoid muscle. That was that sheet of muscle that you learned about in lab. The hard palate, it contains transverse palatine folds that are going to help the tongue when moving the food back towards the pharynx. And then the soft palate has a central projection, which we call the uvula, right? That's your little part right there that kind of hangs out. And then you have the glossopalatine arch and the pharyngopalatine arch, which are going to enclose the palatine tonsils here or here. The tongue is going to be made of skeletal muscle, which means you have conscious control over it. It's also made up of stratified squamous epithelia with different types of papillae for taste buds. Linguinal frenulum, which is going to kind of help the tongue connect underneath. And then the dorsum is the oral portion or the pharyngeal portion where you have the lingual tonsils underneath. You have 3 different types of salivary glands. You have parotid, submandibular, and sublingual, which just means in the cheek, under the mandible, and under the tongue. And they're all going to help produce saliva and drain it into the mouth. So saliva, what are its components, right? It's going to be made up mainly of water and a little bit of solutes, like salts and ions, but it's also going to be made up of enzymes like salivary amylase, lysozymes, which have biotics or microbial action, and then also mucus, which helps add to the viscosity. So salivation actually occurs under autonomic nervous system control. So if your parasympathetic nervous system is activated, you're going to increase salivation, which means it can be activated by sight, smell, sound, memory of food, or even tongue stimulation, like something touching your tongue. And then sympathetic nervous system is going to decrease salivation. Fear causes quote unquote dry mouth, which would mean that you would be less hungry at that point. Okay, so human dentition. We are omnivores, which means we use our teeth to chew up things like meat. We have different types of teeth. Your baby teeth, or your what we would call deciduous teeth, are the kids' teeth that you have 20 of them, and you would lose them all and have new teeth come in underneath. Adult teeth, you should have 32. Though it's very likely that you're probably going to get your wisdom teeth extracted out, so probably end up with something like 28. You'll have incisors that are going to be the middle 2. Then you have canines, which are the ones that are pointed. Premolars, which are going to be just before the molars. And then you have your first molar, second molar, and third molar, which would be the wisdom teeth. The anatomy of the tooth. So if you take a cross section of it, right, it's going to have enamel on the outside, and that's the white part. And then the dentin is going to be just deep to that. And then inside of here, you have a pulp cavity that's going to have nerves and vasculature. And underneath this area, you're going to have the root, right? So the root would be the part that you can't see. the part where the root is going to have the vessels and the nerves exit is going to be called the root canal. And this is all going to be inserted into bone. So this is a joint that we call gomphosis, if you guys remember, right? That basically means these are immovable joints. The teeth themselves are actually bone, so they have the ability to move. And because your skeleton basically rebuilds itself every 5 years, it's not uncommon for you to see teeth shift every very slightly, but every so often you'll notice teeth shifting around in your mouth. Okay, so digestion in the mouth, again, that's mastication. The teeth are going to crush and grind the food. The surface area of the food starts to get increased for chemical digestion. Salivary amylase is going to come help digest the beginning of the carbohydrates. Water gets mixed with it, and then the end product is the formation of a bolus. So a bolus is this like ball of food that had saliva added to it, which is going to begin the process of deglutition, which is basically swallowing. So swallowing is voluntary and reflex movement that involves the swallowing of food. The bolus gets pushed first into the pharynx, and then the larynx is going to rise up. Epiglottis will close over the glottis, and then the bolus can be pushed down the esophagus. That step right there, where we say the epiglottis closes the glottis, basically you're covering the trachea, so you don't. What people would typically say is my food went down the wrong tube, but really what they mean is the food went down the trachea instead of the esophagus, and they ended up choking on it. The pharynx, it extends from the internal nares to the esophagus posteriorly and then to the larynx anteriorly. It's composed of skeletal muscle and it's lined by the mucous membrane. The nasopharynx functions in respiration only, and then the oropharynx and laryngopharynx have digestive functions as well as respiratory. Okay, so deglutition occurs in 3 phases. Voluntary and involuntary are going to be, so you have both conscious and subconscious control, but the 3 phases will start with here for deglutition. So the voluntary phase is when the tongue and jaws push the bolus back to the pharynx. Then you have the involuntary portion where you have the swallowing take place, which again, larynx is rising, epiglottis closes, tongue pushes back, and the food then enters the esophagus. Sorry. Then you have down the esophagus, it connects to the pharynx, to the stomach. The mucosa, the submucosa, and the muscularis layers then are going to be the outer part that we call adventitia. It's going to be lined with stratified squamous epithelia, which is mucoid, and again, that's just for protection. And then the esophagus has 3 parts, the neck, the thoracic, and the abdominal cavity, because it's pretty long. And then it has a collapsible muscular tube, which is going to be posterior to the trachea and the heart, anterior to the vertebrae, and it actually pierces the diaphragm. So you have like a hole that goes to the diaphragm, which we call the esophageal hiatus, so that the esophagus can then meet the stomach through the diaphragm. The abdominal cavity and the peritoneum, it's going to be made up of the serous membrane. The parietal and visceral layers are going to have intraperitoneal and retroperitoneal components, which just means inside and then on the back. The mesenteries are going to be double layered folds of peritoneum that help suspend all of these internal organs in place so that they can stay in position even if you change, right? So like if you invert yourself and you turn upside down, it's not like your organs are going to fall into your thoracic cavity, right? They'll stay in the abdominal cavity due to the mesentery keeping them in place. And then the peritoneal cavity, there's potential space here between the visceral and the parietal peritoneum, depending upon what you're talking about. Again, you have different types of mesenteries, like the lesser omentum, which is going to combine the lesser curvature of the stomach to a portion of the duodenum, then to the liver. I don't need you guys to know what they connect, just that the fact that the mesenteries help adhere all of these things to the abdominal cavity or walls in order to keep them in place. This is just the stuff that sits retroperitoneal of the digestive system. So you have the duodenum, the pancreas, the ascending, descending colon, the rectum, and then also the kidneys that sit on the backside. Okay, so the stomach of the digestive tract is very important. It's basically a place for storage for food. Food again comes in through your mouth, moves down the esophagus, and then into the stomach. Now that happens fairly quickly, right? With swallowing, so chewing, deglutition, and then moving the food. But then here, the stomach can actually hold food for anywhere between 2 and 6 hours. So it really is a storage place. And the good thing here is that you're able to time release food from the stomach into the small intestine, so you don't have um, too much food moving through the small intestine too fast, which would prevent you from digesting all the nutrients out of it, right? You want to allow the food to take its time in the small intestine to be processed through there so that you can gain as much nutrients out of the food as possible before moving it on to the next part. The gross anatomy. So you have different regions of the stomach. You have what we call the cardiac region of the stomach, which sits in this region superior, which basically just means by the heart. Then you're going to have the fundus of the stomach. The fundus is going to be this like upper portion here, which just sits more lateral. The body is the main portion, and then the pylorus is going to be right here at the inferior portion, which is going to basically be leading the food or taking the food into the small intestine, which begins right at this place. You have some sphincters here, which are muscular rings that are going to help allow food in and keep them there. The first one's going to be called the gastroesophageal sphincter, which basically is a muscular ring that's going to attach the esophagus to the stomach. And then the second one's a pyloric sphincter, which is going to open and close and allow food to move from the pylorus of the stomach into the small intestine. And then you also have a lesser curvature and a greater curvature, which are just the way that the stomach bends. And the lesser one is just smaller than the greater one, which is common vernacular for anatomy, right? Lesser is smaller than greater. If you open up the stomach on the inside, like seen in this image, we have what's called rugae on the inside. So these are basically folds of mucosal layers that are going to allow stretching of the stomach, which is great because when your stomach is empty and relaxed, basically it's the size of your clenched fist. But very commonly, we consume food that it would fill up more than the area of our clenched fist. So we have all these folds in our stomach that allow our stomachs to expand. And then they will undergo contractions to help digest food. In the stomach, you have 3 layers of muscle. You have a longitudinal layer, you have a circular layer, and then you have an oblique layer. And this outside one is longitudinal, then you have circular, and then the diagonal one is oblique. And these all help move food, not just through peristalsis, but then also a little bit of segmentation. And then again, you also have muscular rings here, like the pyloric sphincter. Okay, so the submucosa and the mucosal layer or surface is going to be made up of mucus cells. The idea there is that they're going to secrete mucus in order to provide an added layer of protection because the stomach is very acidic. It has a pH of about 2. And with a really acidic stomach, you want to your body has to be very intentional about creating protective barriers because without them, the acid of the stomach would literally just burn holes through. So we have in our stomach what we call mucus neck cells that produce mucus to protect from acid. In the stomach, you have a couple different types of cells. So you have the mucus neck cells, then you have the parietal cells. The parietal cells are going to be to make hydrochloric acid, which contributes to the acidity of the environment. Then they also release what's called an intrinsic factor, which helps with vitamin B12 binding. And then the chief cells in the stomach are going to help by releasing pepsinogen. Pepsinogen is going to be converted into pepsin, which will help digest proteins. And then we have enteroendocrine cells, like gastrin or G cells that release gastrin. And then some other hormones. So there's 15 other hormones like serotonin, somatostatin, and CCK that all help with digestion. Okay, so again, mechanical digestion is like peristalsis, right? It's moving of food. Then chemical digestion is now the conversion of your macromolecule into your like your monomer, which is its simplest form. So for example, a protein into a peptide, right? And you can do that with the use of pepsin, which is an enzyme that helps digest the protein. There are some of these enzymes that are effective in an acidic environment. So they love that the stomach has a pH of 2 because that's where they're fully functional. And then also in the stomach, you create chyme. So because we said these cells, right, create a lot of different components that get added into the bolus. When you move the bolus into the stomach and you add in, we just call them gastric secretions, which just mean all of the secretions from the stomach. Now we're going to call this chyme. So it's this like creamy mixture of food that's beginning to be broken down. And then you're going to have peristalsis occur in waves. So you can move the food from the stomach and like the cardia to the fundus to the body to the pylorus, where you're able to basically squirt the chyme into the duodenum when ready, right? When the pyloric sphincter opens. So absorption in the stomach wall is actually impermeable to most substances. So you can't do a lot of absorption here. You are, however, able to absorb some water, electrolytes, certain types of drugs like aspirin or alcohol through the stomach lining because they're lipid soluble or very small. But that's what it's limited to. After that, we would have to do the rest of it in the small intestine. So the small intestine is the area where you have most of the absorption occur. And that's largely due to the fact that it's very, very long. It has 3 different parts to it. It's about 22 feet in length if you were to pull it out of the body and lay it all the way out. And then also they have what we call villi and microvilli on them, which are these projections that increase the surface area. When you increase the surface area, you increase the rate of absorption, which increases the overall absorption, which is why small intestine is the primary portion of the GI tract to absorb nutrients. When you break it up anatomically, that first portion out of the stomach is called the duodenum. Some people call it the duodenum. It doesn't matter to me as long as you understand it's the first part. Next is the jejunum, and then the last part is the ileum. And that, so it's the yellow is the duodenum, the blue is the jejunum, and the purple is the ileum. And the ileum ends right before you enter into the large intestine down here. Again, the small intestine has specializations to increase the surface area. They have what we call plicae circularis, which is going to be permanent ridges in the mucosa, and it helps enhance absorption by increasing surface area and then causing the chyme to spiral or like rotate as it passes through the small intestine. Then they also have villi, which are going to be the surfaces of the ridged lines with finger-like projections, and then the microvilli, which are cells lining the villi, which we would call like brush border enzymes, which again increase the surface area. So here's a really good image, right, of the difference between them. So, and you can see like you have the plicae circularis, and then if you get a little smaller, you can see you've got the villi, and then on the villi you've got microvilli, which all of them help increase surface area. So when you're looking at the anatomy of a villus and the epithelia of it, you'll call this simple columnar epithelia, which is one layer of tall or column-shaped cells. And again, they have microvilli on them, but they're really helpful for absorption. They also have goblet cells, which secrete mucus there. In the core of the villus, you have a lacteal system, which is going to help with the transportation of fatty acids because they're big. And then also a capillary network, which is going to pick up all the smaller things like the amino acids, the monomers, and like disaccharides and such. Then we have intestinal crypts. So an intestinal crypt, so you have the villus that goes up, and then the intestinal crypt is the one that dips down. This is the crypt of a Leiberkuhn. So it's an intestinal gland where you have goblet cells that release mucus, but then you also have the enteroendocrine cells that release some hormones like cholecystokinin or CCK, secretin, and the GIP, which it's like a glucose like induced phosphate. Then you also have Brunner's glands in here that are going to be submucosal glands, and they release alkaline mucus into the duodenum. And then lastly, we have Peyer's patches, which are going to be like lymphoid nodules in here. So when you look at regional differences, the Brunner's glands are found in the duodenum. The plicae circularis are found in the jejunum for absorption. The villi are very numerous in the jejunum. And then the goblet cells are going to increase from the duodenum to the ileum as needed for lubrication, where they would also have Peyer's patches. And then the goblet cells will be most numerous in the ileum, because this is where you typically have a lot of bacterial load increasing from the buildup of the duodenum and to the ileum. Okay, so mechanical digestion in the small intestine. A lot of this is segmentation, right? And it's the movement, the churning over of it. So you have the ability to have all of the food, whether it's on the inside or the outside of the I guess chyme, you would call it chyme at this point, and it has the ability to interact with all of the epithelia so that you can take the nutrients out of it. Then you have peristalsis, which is going to help the contraction of the longitudinal smooth muscle, and it's going to help propel the chyme forward to keep moving it from one place to another. Okay, accessory organs in chemical digestion in the small intestine is going to be aided by substance from the accessory glands like the pancreas, the liver, and the gallbladder. And exocrine secretions, when they enter the duodenum, they go through what we call the minor duodenal papilla, which is an accessory pancreatic duct that carries pancreatic juice. So whether it's, bicarbonate or hormones, it carries those. Or the major duodenal papilla, which has a hepatopancreatic ampulla that holds the pancreatic juice and bile. And then also the hepatopancreatic duct, which is going to be formed from the junction of the common bile duct and then the main pancreatic duct. The pancreas itself has 3 different portions to it. So you have the head, the body, or the neck. So we'll go head, neck, body, tail. The tail is going to taper off towards the spleen, and this part actually anatomically is not connected to anything. It kind of flaps, if you will. Histologically speaking, the pancreas is made up of lobules of pancreatic acini, and then ductules for the pancreatic juice. The main pancreatic duct is called the hepatopancreatic ampulla, and then the accessory one would be the minor duodenal papilla. It is going to hold and release sodium bicarbonate when necessary to neutralize the stomach acid. So basically what happens is when your stomach stretches, because you're consuming food, the stretch receptors in your stomach will tell your brain, hey, it's time to start preparing for digestion. The brain will then send a signal to the pancreas. The pancreas will release sodium bicarbonate, and sodium bicarbonate will go and literally just sit inside of the duodenum. And it will be sitting there waiting for the chyme to come out of the stomach and into the duodenum so that it can buffer out the acidity right away. Otherwise, the small intestine would be in danger of having holes burning through it. Also, you have pancreatic amylase. So pancreatic amylase is going to also help digest carbohydrates. You have proteases that are going to break down proteins like trypsin, chymotrypsin, carboxypeptidase, lipases that are going to help digest fat, and then ribonuclease and deoxyribonuclease, which will help break down nucleic acids. Then you have the liver and the gallbladder. So numerous functions, from producing and storing bile to emulsifying lipids for that reason. So that would be the gallbladder. So the gallbladder is that little green structure that you see right there. The liver is what actually makes bile, but then it gets stored and concentrated in the gallbladder. So stored is easy, right? It just means it's being held there. Concentrated means you're actually pulling water out of it. So you're going to pull the water out of the bile mixture, and you're going to make it more and more concentrated bile, which basically means you're taking out any excess water and just leaving the bile components. Next, you have liver, right? The liver is going to be to store glucose as glycogen, store fat-soluble vitamins, detoxify the blood, make plasma proteins, destroy old red blood cells, produce urea, and then synthesize and store lipids. So the gross anatomy of the liver, you've got the right lobe and the left lobe. So this side is the left side of the body. So this would be the left lobe, the right lobe. You have the caudate, which is the superior subdivision of the right lobe. And then the quadrate, which is the inferior subdivision of the right lobe, the left lobe itself, and then the porta hepatis, which is going to include the hepatic portal vein or HPV, the common hepatic duct in here, and then the hepatic artery right in there. So the hepatic portal system is going to be include the inferior mesenteric vein joining the splenic vein. The superior mesenteric vein is going to join to form the hepatic portal vein, which enters the liver and it carries the products that are absorbed in the digestive tract. The gallbladder, again, it's going to store and concentrate bile. It has a body, fundus, and neck, and it connects to what we call the biliary tree. The biliary tree is the left and right hepatic duct, which comes from the common bile duct. It also contains a cystic duct in here, and then the common bile duct, which would drain. When you're looking at liver histology, you have hepatic lobules. So we have what we call portal triads. This is where you have like the hepatic artery, the portal vein, and then the hepatic duct, which all come into one area. And they're going to be surrounded by these hepatic plates, which have bile canaliculi in them, which are these little like lakes that are going to help carry bile. Those are going to be surrounded by hepatic sinusoids, which are going to be for dual blood supply. And then the production of Kupffer cells, which help with your lymphatic system. And those are all going to surround what we call the central vein, which you see here in the center. So function, you have lipid metabolism, right? You're going to have hepatocytes that are going to be able to secrete bile into the canaliculi to help emulsify or break down fat. You can break down fatty acids in order to generate or create ATP. And then you can synthesize lipoproteins to transport fatty acids into the blood and start moving things like HDL or LDL, which is going to give you the good and bad cholesterol. So generally speaking, when you say HDL, which is high density lipoproteins, that sounds bad, and low density lipoprotein sounds good, but it's actually opposite. So a high density lipoprotein is actually good cholesterol, and the reason why it's good is because high density means it can handle a lot of weight, which means it can be added to different things and be carried in the body, which means it moves. LDL means it's low density. It doesn't want to get at anything. It doesn't want to move, so it's going to stay stuck. The problem with LDLs is that those low density lipoproteins could build up on each other and do something like clog an artery or vein, which means you're preventing blood flow and circulation and oxygen and energy to that portion of the body. Next is carbohydrate metabolism. So you have liver that's going to store glucose as glycogen. And then you have glycogenolysis, where you're going to break down the glucose. If it's low in circulation, take it back to increase it. Glycogenesis is the creation. So it's when blood sugar is high. The liver will create glycogen from glucose and put it into the bloodstream. And then gluconeogenesis is when the liver creates glucose from amino acids and lactate. The hepatocytes, again, you have protein metabolism taking place here. So you're going to remove an amino group from the amino acid and use it to produce ATP. This area can also convert it to urea for the elimination in the kidneys. And then the last part here is you make plasma proteins from the hepatocytes. You also have the ability to excrete bilirubin, which when built up in your body becomes bad, activate vitamin D, store fat-soluble vitamins, and then also detoxify drugs and other substances, secreting them into bile. So basically what we're saying is if you consume lots of drugs or other substances like alcohol for a long period of time, it could cause something like liver failure because your liver is having to work too hard, too long, and it would render it ineffective, which would be a bad thing. Bile and lipid digestion. So bile is going to be synthesized in the liver. It's amphipathic molecule, which means it emulsifies fat in the small intestine, and it has increasing surface area of lipid globules to allow the pancreatic lipase to break down the triglycerides into fatty acids and glycerol molecules, which are smaller components. From there, you can create this like circular or spherical formation of what we call a missile. And then you're able to start breaking the components down and absorbing them into the intestine now because they're smaller, where they're going to enter into circulation, likely through a lacteal, because even though it's smaller, it's still pretty big. Okay, chemical digestion in the small intestine. So we have major digestive and absorptive functions of the GI tract. And again, a lot of it's in the small intestine. Segmentation and the different motility methods are used to move them. And then all four macromolecules can be chemically digested with the help of enzymes and secretions of the GI tract in the villus. So carbohydrates, again, as a review, you can start the breakdown of carbohydrates, but carbohydrates alone in the mouth because you have the presence of salivary amylase. So if you guys are noting, amylase itself is an enzyme that breaks down carbohydrates. The difference between salivary amylase and pancreatic amylase is literally just where it comes from. So salivary amylase comes from the salivary glands. Pancreatic amylase comes from the pancreas. They both do the same thing, which is break down carbs. The esophagus and the stomach for carbs, there's no digestion occurring there. And then you begin carbohydrate digestion again in the duodenum due to the presence of the pancreatic amylase. And then the brush border enzymes are going to act on disaccharides and start to produce smaller monosaccharides for absorption into your capillaries. Protein digestion, this can begin in the stomach with the use of hydrochloric acid because it has the ability to denature or unfold the proteins. And then also with the use of pepsin, which actually is the enzyme that can break down proteins. From the pancreas, there's digestive enzymes that are going to split peptide bonds between different amino acids. And then the brush border enzymes like carboxypeptidase, aminopeptidase, and dipeptidase are all going to split up the peptide bonds that then can be taken into the capillaries. Lipids, okay, so lipids are very big even when broken down. So lipid digestion occurs largely in the small intestine with the use of emulsifiers like bile and enzymes like pancreatic lipase, which break them down into their smaller forms of triglycerides into fatty acid chains and glycerol molecules. But you don't have any brush border enzymes in this area, so you're going to create the formation of micelles, which is going to help basically repackage the fats and move them across the membrane so that they can be reconfigured on the other side and continue the digestion process. On the other side, right inside of the cell, we would call these chylomicrons. This is where you have protein coating occurring and basically a repackaging of lipids that are going to be absorbed through the micelles. The chylomicrons would contain triglycerides, cholesterol molecules, and lipids, and specifically these would be HDLs. The phospholipid in the protein is going to coat the chylomicron so it can be exocytosed from the intestinal absorptive cell into the lacteal. The digestion of nucleic acids is so minor because by the time you're digesting your nucleic acids, you're really concerned with really starvation for too long and death possibly. So I'm going to go ahead and just leave this out because it's not very energetically favorable, though it, as a last ditch effort, your body could try to break down the nucleic acids for energy if it needed to. Then you would take the remnants of the digestive tract and propel that stuff into the large intestine. So if you're looking here, this is the ileum of the small intestine, and this first portion of the large intestine is called the cecum. So all you did is put those two names together. So ileum and cecum becomes the ileocecal valve, which is going to be the valve that opens and closes to move food from the small intestine into the large intestine. And then the large intestine, when food or material is here, the last job it has is to absorb some water and electrolytes while forming feces. The anatomical regions, so you've got the cecum, the ascending colon, which just means going up, transverse colon, which means going across. The descending goes down, and the sigmoid colon kind of does this like S around here into the anus. Histologically speaking, in the large intestine, you have this band right there that you call the tene coli. Basically, it's a band of longitudinal muscle that helps the contractions occur. You have what we call haustra, which are outpatching, so you can see it kind of bubbles. The bubble itself is the haustra. Then you have epiloic appendages, which are basically fat lobules. And then in the large intestine, there's no villi, but tons of glands. So food digestion kind of goes like this, right? You begin with mechanical digestion of chewing and swallowing. Then you start moving it down the esophagus where you have mechanical and chemical digestion. Then you move to the small intestine where you have basically just absorption, which is going to be chemical digestion. There is still some mechanical digestion occurring. And then the last part here is mechanical digestion or no digestion at all, and it's just moving the food out. So out, right, is elimination. The rectum ends at what we call the anal canal, which is going to be innervated with veins that, when irritated, they could be hemorrhoids. Then you also have an anal sinus, which is full of mucin to ease movement. It's going to be controlled by an internal and external sphincter, which basically means that you have no ability to control this, but a lot of ability to control that. And then also it's going to be lined with the levator ani muscles. Colic flexures. So we have the haustral churning, which in this case movement is slow. So you have lots of water reabsorption occurring. Mass peristalsis occurs 2 to 3 times per day, mostly in the response to gastric and duodenal distension. Gastroileoreflex, which is when gastrin relaxes the ileocecal sphincter to empty the small intestine, and then the gastrocolic flexure, which is when you have a strong peristaltic wave, which is going to move the contents from the transverse into the rectum. And then the defecation reflex is you relax the internal sphincter, which is mediated by the myenteric plexus. You'll supplement with the parasympathetic system, which has conscious control over the external sphincter, which would allow material to move out. Homeostatic interactions. So the digestive system, you can start with the skin and talk about how the intestine absorbs vitamin D, which helps with calcium concentration. Then you go to skeletal. That's where you hold the calcium and the phosphate for the absorption, or through the absorption of the small intestine, you're going to store them there. The muscles, you've got calcium for contractions all over the place so that you can continually do things like peristalsis and segmentation. The nervous system, you're going to get signals from the liver about what to do. On either side, whether it's sympathetic or parasympathetic. The endocrine system, hormones that are going to regulate the digestive system. The cardiovascular system is going to innervate the digestive system to help provide blood supply so it can carry away the nutrients. Lymphatic is going to have things like chylomicron components in the chylomicron system to help with its innate immunity. Respiratory brings so the kidneys assist in the lungs and homeostatic functioning of pH. The urinary system is going to be controlled by the volume, pH, and the content of blood. And then the reproductive system, the male urethra has shared functions both in urinary and reproductive here. Last parts are clinical applications. So fiber, right? Dietary fiber may be classified as insoluble or soluble, depending upon what you're taking. Both types are going to affect food passage through the GI tract. So as long as you're consuming enough insoluble or soluble fiber, material should be moving through your system very well to keep you normal. Mumps are going to be inflammation and enlargement on the parotid glands, which could be caused by a virus. Symptoms here would be fever, pain, swelling, which would be irritable at best. If the mumps are contracted post-puberty, it is possible that they can experience inflammation and, in severe cases, sterility. Okay, let me know if you guys have any questions. If not, I'll see you guys back for our last lecture.